So as we start into chapter 11, just going to talk a little bit more about prokaryotes. There are particular reference materials that you can use to identify bacteria, the taxonomy that's involved in this. Berge's manual is one of those types of references that you can use. Again, I don't expect all of you to rush out to your local bookstore and purchase this, but just understand that those who work with microorganisms, just as we have reference material for other subjects that we study, there are reference materials that help to identify particular microorganisms. We've already talked about the use of RNA sequences in the cladogram, keeping historical data to help determine the particular characteristics of microorganisms. And so a lot of this information that we talk about is collected in the Berge's manual, not only to help identify prokaryotes, but eukaryotes, archaea as well. When we talk about prokaryotes, you do need to be familiar with the typical characteristics of a prokaryote. You should know this. And again, you've already looked at this from the previous chapter, so most of this should be review for you. Prokaryotes basically are one singular or one circular chromosome. It's not in a membrane. If you look at the eukaryotes, if you recall, the genetic information is in a membranous organelle. You don't see a membrane surrounding the circular chromosome. You don't see the organelles as you see in the eukaryotic cells. There are no histones that allow the DNA to wrap around and pack into a tight nucleus. You do see peptidoglycan in the cell walls. It separates by binary fission. Proteobacteria, which include most of the gram-negatives, these are chemoheterotroph bacteria that are assumed to have arisen from a common ancestor, again, as we look at historical information. Few are now photosynthetic. The name derived from a Greek god, if you're into Greek mythology. Basically, most of these are gram-negative organisms. One of the organisms that we do see, even in a clinical setting, is Azosporidium. This is basically in the soil of bacterium. It grows in a close association with roots and tropical grasses. From time to time, individuals can get infected with this organism, though it's extremely rare. It's excreted nutrients and acids in plants, and fixation in the atmosphere is primarily where you'd see it located at. Acidobacter and Gluconobacter, this is a particular genre. We see it industrial importance in these microorganisms. They're aerobic bacteria and will convert ethanol into acetic acid. Rickettsia are organisms that we will see in disease, in various diseases. I will hold you to task to recognize Rickettsia, because Rickettsia is going to be an organism that you're going to see in the system. The rickettsial diseases that we see commonly, that we hear reported, are Rocky Mountain spotted fever. It's caused by a particular Rickettsia rickettsii. It's a tick-borne disease. The organism actually is carried on the tick bites man, and man gets infected. So Rickettsia is one of the microorganisms that would be important to know. Another tick-borne disease that we see is ehrlichiosis, and this is a tick-borne disease as well. Rickettsia-like bacteria that lives in the white blood cell of organisms. Just a little historical data about it. Discovered in 1987, had 250 cases of the disease through Oklahoma, Missouri, and Georgia. The time of greatest risk for this organism is between May and July. And we'll go into a lot more detail when we talk about blood-borne diseases later on in the semester when we talk about microorganisms that affect humans. There's an incubation time, a period of time when you get infected with the organism, that it has an opportunity to increase in its number or grow. The incubation phase before you would actually see disease occur usually runs between 21 days. May occur as early as 7 days. Now, during this period of time, depending on the microorganism that you're talking about, that person may be infectious. Now, in Rickettsia, that's not the case, because it's got to be from tick to tick. But in certain organisms, viruses that you may get in contact with, certain bacteria, strep Pyogenes, for example. These types of organisms may incubate in your system, but at that same time you may also be able to transmit this disease to someone else. Again, now rickettsia is a poor example to using that. That's not how this disease is spread. But there are certain bacteria that will incubate in your body, but you are infectious during that period of time, so other people would be able to pick up the disease, even before you became physically sick from the organism. 20% of victims will develop a rash in urculosis, and then the rash is seldom as you see on the hands and the feet, and the disease may range from mild flu-like symptoms, or a person may lead to severe diseases of high fevers, chills, and may even lead to death. Caulibacter is not one of the organisms that you really need to be concerned about. You basically see Caulibacter in aquatic environments and lakes. Rhizobium, it's one of the agrobacterium, and I will mention it because you'll see it infect the roots of different types of plants, especially peas, beans, and clovers. These are the legumes, the leguminous plants. They cause the formation of nodules and bacteria. They fix nitrogen from the soil and the atmosphere or the environment. Agrobacterium is also an organism that you can see that will infect certain plants. You can induce a bacterial DNA in the form of a plasmid that can result in a disease referred to as crown gall. And bacterium plasmids are used in genetic engineering to produce foreign DNA. So it does have not only agriculture applications, but also has industrial applications as well, because it's used to produce certain other bacteria. Bartonella is an organism that can lead to cat scratch fever. When we talk about cutaneous diseases, we'll bring up Bartonella again. It's another organism that you should become familiar with, again, because it leads to a disease in humans. Brucella, also a very common disease in humans. Well, I should say not an uncommon disease in humans. It causes the disease brucellosis in mammals. It's able to survive the process of phagocytosis. If you recall, phagocytosis may be the process by which organisms land outside the cell. By the process of evagination, the bacteria is brought into the cell, and then lysosomes will attack, and these lysosomes will then bind to the vesicle sac and secrete enzymes. But you can see some of these organisms are resistant to that process. This one is able to survive the process of phagocytosis. It's a contagious disease. It can affect animals, and then animals will then subsequently affect humans. Brucellosis is basically attacks animals. Main threat to cattle and swine, which is where we see the disease transfer primarily to humans. As you can see, it affects other animals as well. Disease is known to cause a contagious abortion, and this is referred to as Bang's disease in certain animals, also known as undulant fever. I would be familiar with the name undulant fever because that's the catch-all for brucellosis in various parts of the world. Severe intermittent fever, it accompanies human infections, or Malta fever, which is less commonly heard of, but it is a human disease originally derived from the island of Malta where this organism struck. Nitrobacter, agriculturally important organism. Again, we talked about nitrogen, the process of nitrification in agriculture is important. This particular organism you can see used there. Woolly bacteria is another common organism that lives in the host. It's an endosymbiosis relationship. If you remember, we talked about that process of sharing between the organism and the host by which the organism is existing. And then the pathogens we already alluded to, Bartonella, which causes cat scratch disease, and then Brucella, which causes brucellosis, are human pathogens that we'd have to be concerned about. Most of the organisms that I help you to focus on will relate to human diseases. I'm not so concerned about plant diseases unless there's a direct correlation between what we see in the agriculture. There's a lot of microorganisms out there. Really the key ones that cause key diseases are the ones that I'm going to point you to. A lot of the other ones, not that they're not important to know, it's just for the purposes of this course, it's no reason for us to go through all the agricultural, bacterial diseases and fungi, because you're never going to see it again outside of this class. I really want you to appreciate the more clinically relevant things. Urculosis, again, we already talked about. Rickettsia disease, primarily rickettsia, Rocky Mountain spotted fever. And then, as you can see, there's also a form of rickettsia praecox, which will lead to epidemic typhus that we'll talk about later on in the lecture series. Wolibacter, you didn't have to know. Caulibacter, which is in lakes, not a big thing. Then the agrobacterium, again, we talked about, that causes the nodules on the plants. I'm sure all of you are very interested in nitrogen fixation in plants. But azospirillum, again, is another organism that you'll see in plants that can cause disease. Again, I'm not going to ask you questions about these. And then the acetic acid generating organisms, Aciobacter.